Did you make that announcement? Nice. Oh, nice. Okay. Cool. Well, no, I'll get it later. I'm sure it won't mind. Okay. Sorry? It flips through, but I could pull it up. Okay, there we go. Yeah, but we'll probably end a bit earlier than that because we're starting slightly earlier. Okay. So, all right, welcome everyone to VanBug. My name is Jesseline. I'm a PhD student at UBC and a VanBug volunteer. Before we get started with the day's proceedings, just a quick note, we are looking for 1 or 2 more volunteers to join us. It isn't a terribly huge time commitment, maybe 1 to 5 hours of your time over a couple of months every now and then. But if you're interested, either find one of us, all these 4 people over here, or Will over there, or send us an email, and we will try to get back to you as soon as possible. If you have any questions, please let us know as well. Okay, so today we are joined by Dr. Derek D, who will be giving our main seminar. But before that, we have a brief student presentation. So Hossein Asghari from the Faraz Hotchis lab at UBC will be presenting on accurate and rapid detection of circular RNA through some fancy math. And he's a research assistant in Dr. Hotchis' lab. And so without further ado, I'll get Hossein to come up wherever he is. Okay, there you go. You were hiding.
All right. Hello, everyone. So today I'm going to talk about the project in where I developed a novel method for circular RNA detection. We're going to see. So I'm going to talk about the method, and we will see some results. But first, let's see why circular RNAs or circRNAs are important. And there are some studies that show that circRNAs are involved in some diseases, such as cancer. For example, in this study, it is shown that circRNAs that are exceptionally stable molecules function as efficient and also function as efficient microRNA sponges with gene regulatory potential have some roles in cancer. For example, in the figure on the right, where you can see the hallmarks of cancer, some of the circular RNAs are shown outside the figure. Some of them are in green, some of them are in black. It means some of them are acting as tumor suppressor or have oncogenic properties. So, as you know, RNA is a single-stranded biological macromolecule which is transcribed from DNA. Exons are the expressed regions of the DNA, and any nucleotide sequence between the exons within a gene is called intron. During the splicing event, which is a process to remove the introns and concatenate all the remaining exons together, the transcripts are created. But alternative splicing can happen, which means that a single gene can have multiple transcripts. So from a single gene, multiple proteins can be produced. Circular RNAs are a type of alternative splicing where the 3' and 5' ends are joined together, and a covalently closed continuous loop is created. Now the question is how to detect the circular RNA from RNA sequencing data. The only way to identify the circular RNAs is to actually detect the reads that are spanning back-splice junction sites. Here in the figure on the left, you can see a linear read that is mapped to a typical junction site in a collinear order compared to the reference genome, but the reads that are mapped to the backsplice junction sites will have a reverse order, as you see on the right. So we are going to use this signal to detect the circular RNAs by identifying these backsplice junction reads from the RNA sequencing data. There are several challenges to solve this problem. For example, since the RNA-seq short reads are split on the junction sites, we might get a very few base pairs on each side, and using this information might be, it's not trivial. Also, from a single transcript, multiple backsplicing events can happen. And finally, circular RNAs are usually of low abundance in typical data sets. To overcome this issue, higher sequencing depths are used, but this requires more resources to process. The existing methods usually are built on top of general purpose high-throughput sequencing mappers. One of the most popular ones is called Circ Explorer. These methods usually have some drawbacks. For example, their sensitivity highly relies on the chimeric reads that is reported by the used mapper, and most of them are resource intensive. So to address these challenges, I developed Circ Miner for circular RNA detection, which has two main stages. So in the first stage, we try to eliminate all the linear reads, as you can see in this picture. Some of them might be junction reads, or some of them might be exonic reads. So we try to rapidly eliminate all these reads, and then focus on the reads that look like that, which we call backsplice junction reads. And if you want to see how it looks like on a circularized transcript, you can see it on the right. So after detecting all these backsplice junction reads, we cluster them together based on which transcript they come from and their breakpoints to finally report the circular RNAs, along with the number of supporting backsplice junction reads. So for the first stage, which was fast linear read filtration, we used the idea of pseudo-alignment, which was previously used for transcript abundance quantification in Callisto and RapMap. This technique is really fast, but it comes with some drawbacks. For example, the way it was implemented in these studies, it did not report the full mapping information, and is only able to map the reads to transcriptome and not the genome. So here we developed the first splice-aware pseudo-alignment method, this state. So as I said, since it is splice-aware, it is able to map the reads to both transcriptome and genome. But there's a priority. We always prefer the reads that are mapped to transcriptome. Afterwards, the ones that are mapped within a gene or fusion genes, and finally, the genomic mappings. So after this step is done, and we got the mapping results from this module, we eliminate all the transcriptome full mappings that we got. And the partial mappings that is shown in the circle are saved to be processed further to finally detect the circular RNAs. So we got the partial mapping from the previous stage, but there's a missing part. So here we try to find that part. We use overlapping seeds with a smaller k-mer size to find this unmapped part. And after we find that, we do some compatibility checkings within the transcript to finally report the circular RNAs. Now I'd like to answer three main questions. The first one is whether the implemented pseudo-alignment module is accurate and robust enough to the diversity of junction reads. And second, is the detection module accurate for circular RNA detection? And finally, how does SIRC-Miner perform in terms of time and memory usage compared to other existing methods? So first, we tested the pseudo-alignment module and compared it with STAR, which is a well-known RNA-seq mapper, on 3 different simulated data sets. As you can see, in most cases, SIRC-Miner outperforms the STAR results, especially in terms of time and memory usage. And in 2 cases, it has higher recall and precision. In the first simulation data set, it has lower precision and recall, and that's because of the multi-mappings. And SIRC-Miner doesn't support multi-mapping. And that's because so for that reason, we get lower recall and precision here. To answer the second question, we generated 2 types of data sets. First one is a positive control data set that only contains circular RNA reads. And then we dilute this data set with some simulated linear mRNA reads to get a data set that looks more like the real data sets we have out there. So finally, at the end, we'll have 2 data sets, and each of them will have a positive control and a diluted sample. Here you can see the results for SIRC-Miner as well as 5 other circular RNA detection methods. You can see the precision and recall for both positive control and diluted data sets. And for each of the methods, an arrow is connecting the positive control results to the diluted results. So if you look further, you can see 2 of the methods are showing robust results. The others usually have drops in precision when we use the diluted sample. But SIRC-Miner in green and SIRC-Explorer are showing more robust results. The thing is that SIRC-Miner has more than 10% higher recall on these simulation datasets. So you might say that on dataset 1, SIRC-Explorer might have, it has actually higher precision, but you should also take this in mind that the scales on the plots are different. So the difference in the precision on dataset 1 is not that big. And when we compare time and memory usage, SIRC-Miner generally has the best performance. And finally, we use some real datasets, where RNAs are treated. RNase is an enzyme that digests all linear RNAs, but it doesn't digest circular RNA structures. This is used to create 2 datasets. First one is RNase which is an RNA-seq dataset with ribosomal RNA depletion method. And RNase dataset has also been RNA treated. RNase treated. There's a term called enriched circRNA, which are the circular RNAs that have at least 5-fold increase in the number of back-spliced junction supporting reads when we look at the treated sample. So here you can see the results on a cell line, which had both before treatment and after treatment samples. In the figure on the right, I showed, so we only focus on the top 100 calls from each method, which has the highest number of supporting reads for it. And then we compare these from before treatment to after treatment, and we check whether the circRNAs that were reported before treatment are also reported in the after treatment sample, and whether they were enriched or not. So if it is enriched, we show it in green, otherwise it's blue. Generally, circMiner has the highest number of enriched circular RNAs that are called from the treated sample here. And again, it has the best time and memory performance among all these tested methods. So in summary, circular RNAs are found to play an important role in cancer. circMiner is developed as a standalone module for pseudo-element and also circular RNA detection. The pseudo-element module shows accurate and robust results. Typically, it can align 1 million pairs and reads in a minute on a single CPU. Also, the detection module has high sensitivity and F1 score and uses less resources compared to other methods. I would like to thank my PI, Dr. Paras Hatch, and all the lab members and my colleagues at SFU and BPC. Thank you. Any questions?
So you talked about how that great multi-math.
Yeah, yeah. So far it doesn't support multi-mapping. Yeah.
What is the fraction of RNA that you write?
So yeah. So that won't be a problem for circular RNA detection. So in the first module that tries to eliminate all the linear reads, when we have a multi-mapping, we only focus on one of them. And if we have a mapping there somewhere in the genome, we are going to eliminate it. This is not going to affect the filtration. It's only going to affect the accuracy. Because when we calculate the recall and precision, we look at the mapping locations. And then if you have multiple packages, there might be some confusion. Otherwise, the read is eliminated. It's not going to affect the detection module. But it's just about the accuracy of detecting where exactly that it is coming from. So yeah, it's just for the first module that does the filtration.
Are there a lot of
Yeah, I think there have been some studies that show that. I'm not entirely sure because I'm not an expert in the biology part. But yeah. Thanks.
Okay. I'll pull up your presentation first so I can read off of it as well. All right. So today we have Dr. Derek D. from UBC from the Land and Food Systems faculty joining us to talk about protein aggregation from prions to food protein nanofibrils. So Dr. Derek D. started out from the University of Saskatchewan where he got his undergrad in food science. Then he moved to Guelph in Ontario where he got his master's in food science. And then he decided to take a really big leap from food science to biophysics instead. And then he did his PhD at Guelph as well in biophysics. After that, he did his postdoc at the University of Alberta. Then he decided to go to the States, Georgia Tech. Realized he didn't like Americans. University of Georgia. And then he came back to Vancouver because Canada is better than America. And today he's going to be talking about the work that he's doing now. I'm just going to switch this over.
Okay, can everybody hear me even if I'm standing over here? Okay, I can yell a little bit louder. So thank you for the invitation. Yeah, so I've been here since January at UBC. I actually was born and raised just outside of Kamloops, BC. For those of you who know what that is, it's about 4 hours into the interior of the province. I actually did my first year of university at Kamloops and then transferred to Saskatchewan. And everybody in Saskatchewan goes, "You're from BC? Why are you here?" So I never really did follow a traditional, sensible route, I would say. As I was explaining to some of the students before, my background sort of biophysics and food science, which if you want to be a professor in biophysics is probably a bad idea. So that's sort of what we established. So what I'll tell you about today is a little bit of that journey. Starting with my graduate studies, I basically was really interested in proteins. And this is like the mid-early 2000s, and the big question was the protein folding problem. How do proteins fold? So that's kind of what sucked me into proteins and what I started to focus on. So I was looking at the folding of pepsin, and I'll just briefly look at that. Basically, it all comes down to what is, how do proteins fold? The fundamental essence of it is the energy landscape that dictates the structure of a protein. So that led me to be interested in energy landscapes. So sort of a really biophysical approach to protein folding. That took me to my postdoc at University of Alberta, where there was a guy there, Dr. Woodside, who had these great tools, optical tweezers, that let you look at proteins at the single molecule level, and it lets you experimentally measure energy landscapes in pretty high resolution. I was in Alberta, the scene of the mad cow crisis, so I was there to study prion protein, which is a protein aggregation problem. That led me to look at amyloid fibrils, which is a specific type of protein aggregate. And so when I started my own lab at the University of Georgia, at this time I'd moved to a food science department there at Georgia. I had very little funding, and I had a fluorescence plate reader. I thought, okay, what can I study that I care about with a fluorescence plate reader? Well, it turns out there's a fluorescence assay that you can measure the kinetics of amyloid fibril formation. So I wasn't really that interested in fibrils when I was a postdoc, but I kind of had this connection from prion protein to fibrils, so I decided to study amyloid fibrils. So, okay, just to back up again, what I was interested in is how do proteins fold? So proteins, we can imagine this, if we strip it down to the simplest case, we can think of it as a unimolecular chemical reaction. Oops, sorry. Where we have we have the unfolded protein that's the reactant, and it turns into the folded product. This can be described in terms of thermodynamics, in terms of the free energy equals the enthalpy. It's sort of the balance of the enthalpy and entropy. That determines the structure of the protein. So the thermodynamic hypothesis is that the native state is the thermodynamically most stable state. So that's how proteins know to get to the native state. And basically, thermodynamics is good for telling us the direction that the reaction will go spontaneously. And this is about the 1970s, this idea that, okay, the native state, it's the most stable, so the protein knows to go to the native state. That really only tells you one piece of the story. The other really important piece of the story is the energy landscape. And maybe I'll use the mouse here. So the energy landscape, it's actually, the important thing here is the barrier in the transition state region. So the activation barriers dictate the folding and unfolding rates. And that has some pretty practical importance. It determines the kinetic stability or the shelf life of a protein. It also determines the resistance of a protein to aggregation, as usually aggregation requires the protein to unfold and form these misfolded states. So the larger that barrier, the less likely the protein is to unfold. Okay. So during my grad studies, I basically focused most of my grad studies looking at pepsin and the zymogen form of pepsin, pepsinogen. So it turns out that pepsin has a very unusual folding mechanism in that it's derived from the zymogen form here with the pro segment shown in blue. But the native active form of pepsin is just this orange part here. Pepsin has been long known that if you denature it, you can't get it back to the native fold again, which is a little bit strange. If we think that the native structure of a protein is the most stable form, then why can't the protein be reversibly folded and unfolded? So it turns out that native pepsin is about as stable as a denatured state. So if you unfold native pepsin, it basically just stays trapped in this denatured state. If you add the pro segment as a separate peptide, so we sort of knew, okay, so pepsin starts out as pepsinogen. The only difference is it has this extra 44 residue peptide attached to it. What does that peptide do? Well, it turns out that it basically stabilizes the whole folding energy landscape. But the key thing that it does is it binds to the transition state and stabilizes it more than it does to the other state. So it lowers the folding barrier, so it catalyzes the folding just like an enzyme would. And it also binds more tightly to the native state, than it does to the denatured state. So it actually shifts the equilibrium to favor the native form. And then if you change the pH, the pro segment basically unbinds, and then you go back to this landscape. But because the unfolding barrier is so big, you end up with a kinetically trapped native state. So it's not thermodynamically stable, but it's kinetically trapped, and that's good enough. So this energy landscape came from ensemble experiments, basically just from measuring kinetics of unfolding and folding at the bulk level. So that's a useful approach for studying native protein folding and looking at intermediates. But we also have to consider misfolding and how these different species bind together when we're talking about aggregation. So there's a complicated pathway, or potentially several different pathways, that a protein could go from a native state. There might be intermediate, a fully unfolded state, to misfolded states, and somehow these combine to give you aggregated structures. And so there are intermediates along this way. And the end states of aggregation could be different. So you could have amorphous blobs, just clumps of protein. Or in the case of amyloid fibrils, this is an example of a highly specific type of aggregation, where you get this highly specific structure. So for my postdoc, I wanted to basically go beyond just looking at native protein folding. I wanted to understand, okay, how do proteins go into the aggregated state? And this is just sort of a cartoon. This was sort of our cartoon understanding of the energy landscapes for proteins, including native folding and aggregation. So this is sort of showing that folding is believed to be on a funneled three-dimensional energy surface. So again, you can have intermediates. You have the native state that's relatively low. But why do proteins aggregate? Again, it's believed to be thermodynamically driven, that these aggregates are even more thermodynamically stable than the native states. But there are barriers, activation barriers to go over to get to the aggregated state. Now, basically, there hadn't really been any experimental measurements of the conformational energy landscape for protein aggregation. So if you look through review papers, you see lots of these sort of cartoon energy landscapes. And just to give a little bit of context to this, so I started studying protein aggregation because there was funding to look at prion protein. We also looked at SOD1, which is involved in ALS. But basically, there are a lot of diseases, protein misfolding diseases, that involve generally a particular protein forming aggregation, and oftentimes that aggregation is specifically amyloid fibril aggregation. There's a lot of diseases. This is an incomplete list of some well-known diseases, including Alzheimer's disease and the prion disease. So this is just an example of amyloid plaques. These consist of amyloid fibrils. And the amyloid fibrils could be composed of individual protofilaments that wrap around each other in sort of a helical form. And the key thing about the amyloid fibril is that it's composed of beta strands that are stacked perpendicularly to the long axis of the fibril. So it's sort of one of the hallmarks that all amyloid fibrils have in common. Now, different amyloid fibrils at their core may have different packing arrangements. They sort of all have this beta sheet structure. So yeah, there was a mad cow crisis in Alberta. So that's sort of what brought me to Alberta to study prion disease. But prion disease affects humans, and it affects a lot of different animals. Right now, actually, chronic wasting disease is sort of a growing problem in North America. There are a lot of human diseases. These may be acquired, or they may have a genetic basis. And the key thing about the prion diseases is that they're basically 100% fatal, and there is no treatment as of yet. So just a little background. Before there was mad cow in Canada, the biggest mad cow crisis in the world was definitely in the United Kingdom. And so basically, this gives an example of sort of the effects of prion getting into the food system. So there were up to, let's see, what is it, 180,000 positive cases in the United Kingdom in their cattle population. So basically, they had to cull their entire cattle population, and it's estimated that 500,000 infected animals may have entered the food chain. So they're able to detect mad cow disease in the cattle, and this peaked, and of course they started culling all of their animals. And then about 8 years later, the incidence of a new disease in humans called variant CJD appeared. So about 8 years later. So that gives you an indication of sort of the latency period of prion disease. So it took about 8 years for it to spike in the human population. And in total, there are about 177, I think maybe 178 cases now of people that acquired this variant CJD and died as a result of it. So we figured out ways to deal with mad cow disease, which is to stop feeding dead cows to other cows. That's one good way to basically cut that spread. In North America, there's a disease in deer, elk, and moose called chronic wasting disease. And so this has been spreading over the past few years, both in farmed and in wild populations. So as far as I can tell, it hasn't entered into British Columbia yet. So that's something that we're watching for. Okay, so we know about these prion diseases. Usually I have it on my slide, but I forgot to note it here. In 1997, Stanley Prusiner got the Nobel Prize for his work on prion protein. And basically, I wouldn't say establishing, but promoting this link that it's actually the prion protein itself, or aggregated forms of it, that are the infectious toxic species of this disease. So it's not a bacteria, it's not a virus, it's not some parasite. So that makes prion very special. That was 1997. Fast forward to now, and we still don't really know. So we know, the structure of native prion protein, and that's in our body. Somehow, the infectious prion, we don't know exactly what it is, but we think it's some sort of aggregated oligomer. Somehow that converts the native prion protein and gets it to form aggregated structures, ultimately ending up in amyloid fibril type structures. And of course, this causes disease. We don't know what the conversion mechanism is. And so this is sort of where I entered my postdoc in 2010, is, okay, we want to study prion protein and how it converts, the earliest steps of conversion. So the problem with ensemble bulk methods is that they suffer from something called ensemble averaging. So if you're looking for some key intermediate that might trigger this aggregation process, it's really hard to characterize that intermediate with NMR or some other solution technique because, basically there's, a huge number of molecules, like an unimaginable number of molecules in a 1 milliliter solution of protein. And so if you're looking for one, basically ensemble averaging just blurs out the details. And so it's hard to focus in on those intermediates, especially if they're short-lived or if they're rare. So what we use instead is a single molecule approach. And the benefits of the single molecule approach, there are lots of drawbacks of the single molecule approaches, but the one thing that makes it worth it is that you can detect these states that would basically be invisible to ensemble approaches. So as I mentioned, Michael Woodside has an optical tweezer lab at University of Alberta, and so that's where I went to do my postdoc. And so, I have a food science background and then a biophysics background, so it was cool for me to think that, oh, you can trap stuff with light. So you can actually use light to trap particles. And so it turns out that, this goes back a long time, that light has momentum. It can actually exert forces on things. And so Kepler noticed this, that the tails of comets point away from the sun. This is the basis for the solar sails. And I thought that this was just theoretical, but I guess there's an actual light sail project. So I guess there's actually a solar sail out there now. It's currently flying, yeah. And I know Bill Nye is somehow involved in this. So that's photons hitting this thing, and it's very gentle, but over time, it'll accelerate. So with optical tweezers, this is basically taking a very high-intensity laser beam, pumping it through an objective lens, and at the focal point, it basically creates a trap that can trap trap dielectric particles. So typically we're using polystyrene particles, but there have been a number of particles that can be trapped with these lasers. And it basically creates what we call a three-dimensional spring that can be described by very simple physics laws, Hooke's law. And so you can trap a particle, and you can move it and apply forces to it. So this is how we can trap a single molecule. Now, proteins are too small to directly trap with these optical tweezers. So instead, what you do is you trap particles. So in this case, polystyrene beads. And so we have two traps. So these are the laser beams. These are the trap particles. And so to get an individual protein, we have to keep the traps far enough apart that one bead doesn't fall into the other trap. So to do that, we use double-stranded DNA just to act as spacers. So we call them DNA handles. And then what you do is you put your molecule of interest in between those DNA handles. So there's some bioconjugate chemistry that's required to make up these samples. It's actually one of, sort of a major bottleneck to some of these things. Anyway, so you can get a single protein trapped in there, and then what you do is you pull on it. And again, we pull on it, and we can measure the force as we're moving the traps apart. And so we generate what we call a force extension curve. So we pull on it, you see the protein unfolded. So what happened was, as we pull on it, the DNA handles tighten out. And so this is the DNA handles stretching. And then at some point, at some force, the protein can't take it anymore, and the protein comes apart. And then we just keep pulling out the unfolded protein and the DNA handles. And so this is the force, this is the extension. That's what we call it a force extension curve. If you kept pulling on this, eventually the DNA would start to pull apart at about 65 piconewtons. And you'd have to pull into like the upper hundreds of piconewtons to actually break. But before we get to that high, because we would like to, it takes a really long time just to find a single molecule. So that's kind of one of the major disadvantages of the approach. We don't want to pull it that hard. What we want to do is we want to relax it again, and we can watch the protein refold. So everything relaxes, and at some force, usually lower than the unfolding force, the protein refolds, and then we can pull on it again. And ideally, we would do this over and over and over again and get as many curves as possible, because that improves our statistics. So this is an example of hundreds of force extension curves overlapped on each other. We can just, this comes from physicists, and physicists like to be able to describe everything in as much more detail as possible. We can basically, our goal is to be able to account for like every inch of that data. And so you can fit most of your sample is just double-stranded DNA. And they understand the behavior of double-stranded DNA very, very well. And so you can fit this with a model. It's called the worm-like chain model. We can fit the data before and after the protein unfolds. Because again, without the protein molecule there, we would just be seeing we'd just be seeing this blue curve here. You're just stretching DNA. And so anything that's happening other than this worm-like chain is the protein doing something. So keep that in mind. So we can fit this worm-like chain model. The distance between these two curves gives us the contour length change. And so that's basically the length, the protein started, it was globular folded, and then we stretched it out. That's the contour length change. And we stretch it all the way out. We get the contour length change. This is the main measurable. We get the contour length change, which we can relate to the number of amino acids. So how many amino acids were in that thing that just unfolded? With native prion protein, we have its NMR structure. So we can basically estimate what the contour length change should be for this protein and compare it to our experiment, and they match. So we know, okay, this is the unfolding of native prion protein, or the refolding of native prion protein. The other thing we get that's very important is the force. So basically the unfolding force is, what was the force right before the thing, before this rip? By the way, we call this a rip or a sawtooth rip. So you get the unfolding force. We can also, basically by integrating the area below this curve, we can get the energy that it took to unfold the protein. We can get thermodynamics from this. We can also get kinetics, as I'll show you. So when I started this, we wanted to look at aggregation. So we had data on just a prion protein by itself, just a prion monomer. And so we wanted to go one step further and say, okay, what happens if we put two prion proteins beside each other? And there was some relevance for doing this because there was some biological data to show that dimers were perhaps relevant to toxicity. And so I've been showing you native prion all along here. So this is the unfolding and refolding of just a monomer of prion protein. So a single transition which indicates that there are no intermediates. It's just either natively folded or unfolded. That's it. Two-state transition, pretty simple. So people have done similar experiments with other proteins. And so if you just make a tandem repeat, put two of them beside each other, what you would expect to get are just basically two rips just like that. You'd expect one domain to go and another domain to go. We didn't see that when we put two prion proteins together. So instead, we saw some completely different behavior. So just immediately, this tells us, so we have this rip here at lower force. The contour length changes I'll show you is also much bigger. And then we have something going on up here, and then another transition. And so we can just tell by looking at this right away. You put two prion proteins beside each other, they interact and do something. So we don't have this cartoon. We have this cartoon. I don't know exactly what the structure of it is, but it's doing something different. So the next few years of my life were basically collecting this data and then trying to figure out what it means. So I'll try to skip through it as fast as I can. But basically, when we pulled on this dimer over and over again, we noticed only two different behaviors. We call this one with the big rip and one without the big rip. And so actually, this was a little bit surprising to us because we thought, okay, if we're seeing this folding, and this is two proteins stemming together, so technically it's aggregation. From the landscapes, how people imagined it, they imagined it to be very messy, that like maybe every time you pull on this, you'd see something different. Like maybe it's just random and it's doing something different every time. So actually, at this point, we thought, oh, we're getting two behaviors. And then on further analysis, we realized, like, no, this is actually just one behavior. So, basically, the challenge here is, okay, so we have these force extension curves, and we're seeing rips, we're seeing transitions. Let's characterize all of these states and fit the data with the worm-like chain model. Again, the worm-like chain can describe the behavior of the DNA really well. So anything that worm-like chain can't fit is the protein doing something. We sort of go through this, and we say, okay, there's a transition up here. And by the way, so this at high force, this is the unfolded state. So counting in from the unfolded state, we have this intermediate here. We can fit it. We get a contour length. We can estimate the number of amino acids that are in that intermediate. We go to the next transition that we can see very clearly in this curve with no rip, with no big rip. There's sort of a shoulder here. We call that another intermediate. We call it intermediate 3. We have the contour length. And then, of course, there's the big rip, which we call the misfolded dimer. So that's the most structured this thing is. And so we get the contour length and the number of amino acids. And then there's actually another intermediate in here. And I'll kind of skip over the details of how we identify all these intermediates. But suffice to say, again, that there's a deviation from the worm-like chain. And so that's the protein still doing something. So between the intermediate 3 and intermediate 1, there's still some unfolding going on. And we think that that's intermediate 2. And so we can get the forces and we can get the contour length changes for all of these intermediates. The other reason that one reason that we know that we're actually looking at a single pathway instead of two pathways is that if we unfold it and then let it refold, this is basically what the refolding curves almost always look like. It sort of just retraces the force extension curve, showing us these intermediates. And when we pull on it again, sometimes we get the big rip, sometimes we don't. What we found is the longer we wait at 0 force before we pull on it, so we call that the waiting time or the refolding time, the more likely we saw the big rip. And so what it means is that as we sit down here and wait at low force, eventually this thing folds into that completely structured state, which we call the misfold state. So we can get the kinetics for this process. We can also get the thermodynamics from this. But so basically, and we had one refolding curve that retraced through all of the intermediates. We can see it starting unfolded, goes to intermediate 1, intermediate 2, intermediate 3, and then down here, just a little blip, it goes into the fully structured. That was one curve out of thousands of curves that actually showed that. We didn't need that curve. It was just nice to like, oh, there's one that does it. So we have one pathway with 3 intermediates. We've got the fully unfolded state, and depending on how long we wait, sometimes we come out and we see the big rip. And if we didn't wait long enough and we start pulling on it, we just start from intermediate 3. So this actually was just a total lucky accident on our part. We didn't intentionally do it this way. But we realized we were really lucky because if we had done the experiment a little bit differently and we always, say, waited long enough, we would generally come out of the misfolded state. And when you come out of the misfolded state, it skips over intermediate 3, so it would be invisible to us. So it was really kind of convenient that under our conditions, it was sort of a slow conversion from intermediate 3 to the fully misfolded state. That meant sometimes we would see that intermediate. But anyway, I was just sort of walking you through like how we can get detail out of these force extension curves. One thing is we never saw a native transition when we had a dimer. We never ever saw a rip that looked like a natively folded prion protein. Well, that's kind of special because people have done these experiments with other proteins and they didn't see that behavior. So what's happening here is that this intermediate in the dimer forms at such a high force that we think what's happening is the formation of that intermediate blocks the native folding pathway. And it forms at higher force, so it forms before native folding can happen, and it just takes it down in this folding pathway, and it can never go to the native state without completely unfolding again. And so this intermediate, this first thing here, we think is very critical to the misfolding pathway. So just to back up a little bit, how we make our dimer is we take a prion protein and we covalently link it to another prion protein such that the C-terminus of one is connected to the N-terminus of the other one. So it's an NC-NC dimer. And so we know that this intermediate corresponds to something of about 50 amino acids, and the formation of the structure blocks native folding in both domains. So we think that it has to be forming in the region spanning the C-terminus and the N-terminus of these two. And so you can take those 50 amino acids and you can start doing modeling to try to come up with what is the structure of this intermediate. Well, I'm not a modeler, but I just put it through this server program, Quark, and came up with a model that seems sort of plausible for explaining what the structure of this intermediate. So for now, I'm calling this is the structure of intermediate. Not proven, but that was just a model that we put. Okay, so I was really interested in energy landscapes. This is sort of what brought me to this lab to do this kind of experiment. And you can get energy landscapes from this data. So you can get a lot from these force extension curves. So I mentioned that you measure this thing hundreds, actually thousands of times. You pull on it, relax it, pull on it, and you measure the force of each of those rips. And for each intermediate and the fully misfolded dimer, these are all of the unfolding forces. So we can get a histogram of all of these transitions. We can get a distribution of unfolding forces or refolding forces. This is kind of what's special about the single molecule technique. With the ensemble approach, you'd only be able to get the one average value. With single molecule, you can get distributions. And there are physical models to fit these distributions, which give you information like the activation barrier, folding rates, and distances to transition states. And then again, by integrating the area under the force extension curves, we can get thermodynamics. So we can get the Gibbs free energy changes between the ground states. We get kinetics and thermodynamics. We can get structure from the contour length changes. So with this information, we can piece together the energy landscape for this misfolding of the dimer. So we had previously done the energy landscape for just the monomer prion protein. And again, it's just a two-state. There's native, there's unfolded, there's no intermediates. This is the transition state. The dimer looks quite a bit different. So again, we have these different intermediates. There's some barriers here. This intermediate is somewhat similar in stability to the fully misfolded thing. The one key thing here is that the barrier going into first intermediate 1 is basically almost non-existent. It's very small. So it's almost barrierless folding into that first intermediate in the case of the dimer. That was curious because the folding rate for the dimer going into this first intermediate was actually 5 times slower than the observed folding rate for going into the native state. We thought, okay, how can this be? How can the folding actually be slower if there's no barrier? We're used to thinking of barriers as determining the rates. But now with ensemble approaches, you might use something like Arrhenius. You might remember this from like Caylus-Menten or thinking about how enzymes work. We usually focus on the activation barrier, and it is very important. It depends, the rate depends exponentially on the barrier, so it is important. But there is a pre-exponential term, and with ensemble approaches, we tend to ignore that pre-exponential term. With single molecule, we can use a theory that's considered to be actually more accurate for describing folding, which is called Cramer's theory. And you can get something out of there called the diffusion coefficient. So we measure this with optical tweezers, the diffusion coefficient. And it turns out that for misfolding, the diffusion coefficient is about a factor of 3 smaller than for native folding. This is why the dimer misfolding is actually slower, is because the diffusion coefficient is a lot slower. And this is something that's actually expected for proteins that haven't evolved to fold to those structures. So presumably prion protein didn't evolve to fold to this structure, and so you expect this diffusion coefficient to be slower, because what it indicates, a slow diffusion coefficient indicates more roughness, microbarriers on the landscape. So there's, we think proteins have evolved to be minimally frustrated, so aggregation should be frustrated. And so we were a little bit surprised that we're only seeing this one pathway, but this slower diffusion coefficient kind of matched our expectations with what we would expect for aggregation. Because at first I kind of thought, oh, this protein has like evolved to do this. That's a little scary. And it is prion protein. We could also, this was actually quite new to measure the diffusion coefficient for misfolding. That hadn't been done before. And it is dependent on that Crommer's theory. So some people came at us and like, I don't know if we trust that. But we can actually directly probe this by, instead of ripping the protein apart, we can hold the protein at a constant force and watch it hop back and forth between different states. And what we can measure is something called the transition path time. And so when we talk about rates on an energy landscape, the rate, or the waiting time, which is inversely proportional to the rate, it also includes all the time the protein sits there doing nothing in the ground state. Like it's wiggling and jiggling, but it's just in the ground state. And so Peter Wolness describes this as long periods of boredom. The protein is just sitting in the ground state, and you're waiting for it to hop over the barrier. And that's the exciting part. What is the transition state? That's what people have been going after for so long. So with optical tweezers, we can actually directly measure just that time of like, okay, so the protein is doing nothing, it's in the well, but how long does it actually take for it, once it's going to go over the barrier, how long does that actually take? And that transition time is related to the diffusion. It's inversely proportional to the diffusion. So we can measure that directly. Here's the native protein, so it's just hopping between native and unfolded. And the extension changes, so native, unfolded, unfolded, with native. And basically the native protein folds so fast we can't even measure it. So it's below the instrument resolution, so it's less than 50 microseconds. Again, the diffusion for this is 1,000 times faster than for the dimer from our energy landscape. So if we directly measure it, turns out, yep, it matches with the experiment, because here's that intermediate, here's it unfolding, and we can observe it happening on milliseconds. These are just other transitions, for example. So it's happening very slowly. Average transition path time is about 0.5 milliseconds. So that's model independent. We're just sitting there watching it hop back and forth and actually seeing it go through the transition state, which that has never been done before. Okay. I'll just briefly mention this one. So that was prion protein. That's looking at misfolding with optical tweezers. Again, with native prion protein, we just saw two-state transition, native or unfolded. This is just an example to show you that a different protein can behave completely differently. So this is SOD1. This is involved in ALS. It's believed that the misfolding of this protein is somehow correlated with ALS. So with this same sort of study, just put a monomer of SOD1 and pull on it, and what do we see? So sometimes you pull on it. We always see at least one intermediate. You let it refold. This time it refolded. We saw 2 intermediates. Pull on it again. This time we saw 3 intermediates. And we do this over and over and over again. And it turns out we always see at least one intermediate, but not the same intermediate. And so this is just a histogram showing how many intermediates we generally see. So most of the time, you always see at least one, usually 2 or 3, sometimes 4, rarely 5, and almost never more than 5 in any given 4-6 inch year. But again, you do this over and over again, thousands of times. And so we did this thousands of times with 12 different molecules, and we just pooled all the data together because we're like, how many intermediates are we seeing? And so you pool the data together, and we could identify at least 8 different intermediates based on the contour linkage. And so I'll just gloss over this for the sake of time because I want to get on to other stuff. But this is just showing that this is just monomeric SOD, and its unfolding, refolding pathway is way more complicated than prion protein, which was just native and unfolding. You couldn't detect any intermediates. With this thing, we're seeing 8 intermediates. And so we're able to map out the unfolding, refolding pathway for this protein in pretty much unprecedented detail. You wouldn't be able to do this with any ensemble method, or I challenge anybody to do it. So this is going from the native state for all these intermediates to the unfolded state. The thickness of these arrows indicates the probability that protein goes down that path. So this is kind of like matching with how people imagine folding funnels, that you have different flow channels that a protein could go down to get to the native state, and some might be favored over others. So that's sort of what we're seeing here. To go a little bit further, because this is going from native sod to unfolded sod, we have the crystal structure for this. So unlike the prion dimer, I don't know what the structure of that is. For this, we have a structure. And so we can kind of look at the contour lengths and look at the native structure and be like, what parts of the protein might be explaining these transitions? And so we're able to propose a model that's consistent with the data that sort of shows that you're peeling these beta strands off, and those are the intermediates as the protein unfolds. Okay, so that's what we're able to do to look at protein aggregation at high resolution with optical trapping. To sort of bring it back to me here at UBC, I just put in a CFI this month, and I'm hoping to get an optical trap here on campus at UBC, so that I can continue to do this kind of stuff with proteins relevant to food and health. It would be pretty unique in Canada and probably in the world, assuming nobody else has beaten me to it. So that was, SOD1 and prion. Those are examples of disease-causing protein aggregation. For the last, well, I will say, let's say on the order of like 20 years now, people have been documenting that there are actual protein aggregation and amyloids that seem to perform a beneficial role to an organism. So these are called functional amyloids. So they're found in yeast, bacteria, biofilm formation. This is a particularly relevant one for me. It's relevant to food safety, so that's one that I'm looking to go after. It's also very interesting because it's a regulated, well, these are probably all regulated examples of regulated amyloid fibril formation. So here's a bacteria basically shooting out curly amyloid. It's also interesting because this is the kinetics of curly amyloid formation. So it's mainly the CSGA protein that forms the amyloid, but there are other proteins, so CSGB, that catalyze the amyloid formation and other ones that inhibit the amyloid formation. It would be interesting to understand the mechanism of how these proteins either catalyze or inhibit amyloid formation. There might be some applications. There are also functional amyloids found in humans, so for catalytic scaffold and for hormone storage. And the list of functional amyloid is growing as more people get into this area. So in addition to natively occurring functional amyloid, people are interested in making amyloid and engineering it as a novel nanomaterial. And this sort of gets us closer to the food science applications. So amyloid fibrils have unique properties. So they're long and skinny, which gives them a high aspect ratio. They also have interesting strength and elastic properties. So this is the elastic modulus. The amyloid fibrils that have been characterized span more than 2 orders of magnitude. So that shows that this could potentially be engineered. It has very tunable properties, as well as strength. Interesting surface chemistry, but compared to other nanofibril type polymers such as cellulose, proteins, 20 naturally occurring amino acids, and then maybe like 100 unnatural amino acids that we could use to get unique surface chemistries. So there's an interest in using these. First, most of the research has been done in nanotechnology, but also in food, for the functionality in food that nanofibrils could provide. And so this is an area that I would say since about 2010 seems to be growing quite rapidly. So if we want to intentionally make amyloid, and it's tunable, so we can get different strength, different elasticity, different surface chemistry out of it, we need to understand the mechanism of how amyloid forms from the starting material, which is generally a native protein. And so again, we've got a long way to go. Amyloid fibrils, I'll just touch a little bit on their complexity. You can get different polymorphs by making fibrils under different conditions. So you could get packing polymorphs, so just the packing side chain interactions are different, or you get segmental polymorphs where it's just different regions of the protein forming the fibril core. So imagine a beta strand fibril core, and then you've got the other bits of the protein that are solution exposed. are exposed to the solvent. And so we can vary physical parameters to perhaps engineer particular polymorphs, because a particular polymorph ought to have different functional properties. So it turns out that you can make an nanofibril, or sorry, an amyloid-like fibril pretty easily, or I'm sorry, calling them nanofibrils. For food applications, I like to call them nanofibrils. Anything that basically mildly destabilizes the protein might be a good way to make a fibril. There are different pathways for making a fibril. Somehow you want to denature it, or you can completely unfold it. And then the two major branches here are either you make the fibril out of completely intact protein, or you do it under hydrolysis conditions, so low pH, with an enzyme that cuts it up, and then it's actually the peptides that form the fibril. And so, as an example, we can do this with lysozyme. So here's intact lysozyme. Here's lysozyme after it's been at pH 2, 65 degrees. And on the order of 10 hours, you get these really nice amyloid fibrils out. And so when I was at the University of Georgia, again, we had the fluorescence spectrometer, so I could do THT kinetics. We also had a microwave, so we could microwave it. And so I said to my student, like, okay, microwave the fibrils and see if that does anything, and measure the kinetics. So it turns out the microwave heating gets you fibrils faster than dry bath heating. That's sort of interesting, but it also is interesting that it looks like we're getting different fibrils based on how they look under a microscope. I think we have fibrils with potentially different properties. I think the microwave is just heating up the hydrolysis of it, because that's why people buy these microwaves, is to hydrolyze their proteins. But so it's doing it faster, but it also may be giving a different band profile. So you may actually be generating a different peptide that then goes on to form the fibril. So it's just one example of how you might control the assembly of the fibrils. We also did a study looking at different polymorphs. So lysozyme has been studied for a long time. There's like 10 different conditions that are known to make amyloid with lysozyme. So one way is just to heat it up at low pH, generate peptides, that get hydrolyzed, and then those go on to form the fibrils. The other way is to do it closer to neutral pH, but you have to have some sort of chemical denature. So in this case, 3 molar guanidinium. And the key difference here is that in one case, you're getting fibrils that are made of peptides. In the other case, you're getting fibrils that are made of intact protein. So just by that alone, you ought to be getting different polymorphs. And so we think that we do get different polymorphs. The fibrils at pH 2 look different than the ones at 6.3. And then we thought, okay, so the other thing that's cool about amyloid fibrils, it's not cool in disease, but it might be cool for nanotechnology, is that they can seed the formation of more amyloid fibrils. So we take the pH 6.3 fibrils, and we use them as a seed for native lysozyme, but at pH 2, 65 degrees, and we say, what kind of fibrils do we get? We think we get a different kind of fibril. It looks like it might be more similar to the pH 6.3 fibrils than it is to the pH 2 fibrils. So this would be kind of cool because it means that we could make a seed under one condition and then use it under a totally different condition to get a fibril, but that still has the properties of that seed. And we also looked at, okay, so the seeding phenomenon. It would be a bad thing if you put amyloid fibrils in your body and they seeded formation of your proteins to form amyloid. So we just did a quick check with egg white lysozyme, can it seed human lysozyme? The answer is yes. Under these conditions, pH 2 or with guanidinium. And it turns out that pH 6.3 is a better seed even at pH 2 than pH 2 seed. So this 6.3 thing really wants to seed amyloid fibril formation. We treated these fibrils with Pepsin just to see if they would survive gastric fluid. It seems like they do. So these are the fibrils. They're getting sort of a haircut from Pepsin, but the fibril is still intact. This is cross-seeding under conditions that you would not find in the human body. So if you do cross-seeding under conditions that might be more similar to the human body, you basically get only 1 out of 37 samples showed any increase. This is kind of like an area that we're going to explore more, but most of the research on nanofibrils for food application I would say 99% of the work has been done with milk proteins. So this research goes back probably to around the year 2000 or somewhere around there that they noticed that you could make fibrils out of milk proteins, and a lot of work on that. We make these really beautiful fibrils out of beta-lactoglobulin or egg white proteins, but just recently, people have been doing this with plant proteins. So I have a project right now looking at doing this with plant proteins. So seed storage proteins, predominantly from legumes. So here's an example from soy. And so the idea is, can we make nanofibrils out of these? Can we define the structure-function relationships? Can we engineer them? And are they of any use in food? And then on the same token, looking at the safety of it. Now, just based on sequence similarity alone, plant proteins ought to be more safe than dairy proteins or egg proteins, just because we don't have proteins in our proteome that are similar to those seed storage. So lower similarity ought to be safe. Okay, this also taps into something that's happening right now, and I've been in food science since 2001, and it's kind of blowing me away that there's all of this interest in plant proteins now. We don't have it in Canada yet, but the Impossible Burger, we have the Beyond Burger in Canada. Being in Georgia, I had both of them, and I'm like really waiting for the Impossible Burger. Anybody had the Impossible Burger in person? Like it better? The Beyond Burger? Yeah, so did I. Okay, so anyway, I can't believe how much focus is going into plant proteins now. And so people are doing it for sustainability, for health. You know, there's questions about whether that's good reason or not, but definitely for sustainability. There's a lot of reasons to switch to plant proteins. And so basically, you know, how can we make something better out of plant proteins? And nanofibrils are like one way that, like, you can dramatically change the structure of that seed storage protein into something totally different that has a fibrillar shape. Well, you know, these amyloid-like fibrils. So in Georgia, I started doing this with peanut proteins just because I was in Georgia. They grow a lot of peanuts, and nobody had tried this with peanuts before. So we said, oh, let's see if peanuts can make fibrils. And we just take a crude extract of peanuts, just grind them up, and then put them at pH 2 and heat them up, and we get these really nice fibrils. And there have been another 2 reports of people taking legume proteins and making fibrils out of them. Our fibrils look different. So again, this just illustrates, and this is very early stage stuff. So what are the mechanisms? What are the differences? So, you know, just looking at it, I think our fibrils look different for some reason. And so it's worth comparing the functionality of these fibrils from we're looking into this. And so basically, you know, I went down this path of looking at amyloid fibrils. It started out, everybody was interested in amyloid fibrils because of protein misfolding disease. And then more people are turning their attention to functional amyloid, and then also engineering amyloid, and potentially intentionally adding them into food as food ingredients, or perhaps they're already being formed in some food process. That's where we're going in our lab. And thanks.
Any questions?
Yep.
Looking at the graph, not your thermodynamics and genetically there endpoint. And are there any experiments on like what the temperature impact is? One way.
There could be. I don't know of any, just because of how the instruments were set up. Usually, like say for example, in the lab at University of Alberta, we had no way of changing the temperature. The temperature was just fixed. So you can do it. So somebody may have, but in general, like what will happen is the same thing as if you did it at the ensemble level. It's just going to shift the landscape to favor one thing more than another, right? So you would expect it to shift it.
Yeah, so you're coming from like a more chemistry or also
Yeah. Yeah. I don't know. Off the top of my head, I don't know that I've ever read a paper that was like, here's the temperature effect, but done at the single molecule level. But I would just, so I don't know if there's anything that we would anticipate to happen that would be unusual. I think just from the basic thermodynamics, you might expect it to shift in a certain way. Like if you take the average of your single molecule experiments and get, say, the delta G from your single molecule experiment, it ought to agree with the ensemble measurement. And generally it does. Like the delta G that we measure matches the delta G somebody might measure, you know, using any bulk technique. Sorry? Oh. People are working on getting crystal structures, well, not exactly crystal structures, but fiber X-ray diffraction structures, getting like high resolution X-ray structures from fibrils. Yeah, I mean, there are a lot of people, well, I know there are people actively working on that. There's a guy at University of Alberta that's doing that. And so there are X-ray structures of, you know, be hesitant to say what's the highest resolution, though. It's not down to atomic resolution. But yeah, there are people getting X-ray structures of these.
I have two questions. That is, I'll start with the question. I'm just curious what your sort of expansion curve, the software you use now in area of development.
Oh, so the measurement software for the optical tweezers. So as far as I know, most people, so these are home-built instruments, so the ones that I'm showing here. As far as I know, everybody uses LabVIEW.
And to go from the reputation.
Yeah, and so that you use whatever your favorite program is. So for me, I was using everything was 764. Some people use Mathematica. And then the instrument that I'm looking to get, so I'm not a physicist and I don't want to build my own trap, but so in the last few years, there's a commercial trap that's come on the scene since 2016 that looks like it's able to, it's not as good as the homemade instrument that I'm showing you, but it looks like it's pretty close. And that's from a company called Lumix. And then they have their own software that they've developed to make it a little bit more push button. The whole point is that to get this, like right now, the only people that have these instruments are generally people with physics or somebody that has enough money to hire a physicist.
The sort of multi-science question. What's the reason for using packaging? More environmental.
Oh, well, food packaging is definitely a potential application. I'm not personally looking at food packaging, or not yet anyway. But
is there any properties that make it?
oh, well, so just you know, based on just even their aspect ratio alone, like they might be good at increasing viscosity. They can form films. They can form aerogels. People are using them to form oleogels. And so I would say it's just very early stage right now. It's just that you're getting a completely different property out of the same protein. Going from globular to this thing, and you know, to this long fibril. And so yeah, you can form thin films, gels, stabilized foams. Stabilizing foams and emulsions is like a big deal. You can get something that stabilizes a foam really well at a lower concentration. Somebody might be interested. And then also texture, like with these plant proteins, like right now, people are using texturizers. So they're using an extruder to just take a slurry of globular seed storage protein, high temperature, high pressure, put it through a screw, extrude it. We don't know what's really happening, but we know that we get something out the other end that has a better texture than what we put in. That's called texturized vegetable protein. That's the basis for like Beyond Burger and Possible Burger. So that technology's been around since the 1950s. They've improved it in a recent decade. Nobody knows what the structure of the proteins are once you do that. That's an open area.
I have a quick question. So you talked about how you can build these certain aggregates with older proteins, start with Cs, or are there certain proteins that will definitely never get folded aggregated? Like, are there proteins with, like, for example, I'm thinking proteins that naturally dimerize on an actual function of the cell, will they never be able to go down to certain, or is it possible for them to fold down?
Yeah, the latter part. So, I mean, I would never say, and for any of the review papers that I can remember in my head from, like, you know, the biggest people in the field, like, the quote is, like, like I say, you know, I said or I wrote it, like, potentially any protein could form an amyloid fibril. I don't know of any protein that's been proven. First of all, you can't really prove that it could never form an amyloid fibril, so it couldn't really be proven. But so potentially, if you find the right condition, we think it could form an amyloid fibril. And what we know is amyloid fibrils have been made from, like, a huge variety of different proteins. So right now, my estimation is perhaps any protein with a peptide, polypeptide backbone could form a, so by definition, it seems like almost any protein should be able to form an amyloid fibril. Now, again, under some conditions, we're hydrolyzing it. So like out of 150 residue protein, maybe it's just like a 10 residue stretch that's actually forming the amyloid fibril core. So in that context, not the entire protein, but parts of it should form a fibril.
So with that, let's please join me in thanking Dr. D for joining us tonight. Okay, and since it's Halloween, please join us over in the lunchroom for some Halloween candy and some beer to go with it. And we'll probably have some pizza as well for those who don't like candy.
I just didn't want to forget my USB.
Oh, yes, yeah, that's a good idea. I had moved it over already. Oh, so you can eject it now.
Oh, okay.
Yeah, so it's all good.